Assalamu alaikum dear students, welcome to Dr. Rumi Lectures. Today we are going to study past UQs from chapter 50 and 51 of Guyton Physiology. Our very first question is list the three components of accommodation, describe the mechanism of change in the shape of lens during accommodation. Accommodation is also called as near response. This is the ability of our eyes to keep the image focused on the retina as the distance between eyes and the object varies. For example, if you are looking at a distant object and you have to look at a near object, then all the changes which occur in your eyes, they are called as components of accommodation. The three important changes that occur in our eyes: number one, our lens become convex and its power increases. So it means for near vision, we need more power of our lens. That's why lens becomes globular. Pupil constricts, which means meiosis occurs, and eyeballs converge medially. So it means for looking at a distant object, our eyeballs diverge, and for looking at an object which is placed near, for example here, then our eyes converge. And this is because of contraction of medial rectus muscle. Next part of this question is describe the mechanism of change in shape of lens during accommodation. Lens becomes globular because of contraction of ciliary muscle. Ciliary muscle is like a sphincter and is present all around. When this muscle contracts, it moves forward and it decreases the tension on the zonular ligaments or the suspensory ligaments of our lens. Because of less tension on the edges of lens becomes globular because of its elasticity. Parasympathetic nerve fibers which are coming along the cranial nerve number 3 stimulate the contraction of ciliary muscle. Next question is what changes occur in eyes when these are focused on a near object? Once again, this is accommodation. Explain the nervous mechanism involved. Another question is a man focuses his eyes on a near object. Give changes in his eyes during this focusing. Once again, accommodation. What is the nervous pathway of these changes? There are two important questions for which the nervous pathway from physiology of vision may be asked in your examination. One of them is the pathway of light reflex and the other one is the pathway of accommodation reflex. The pathway of accommodation reflex is that light stimulates the ganglion cells, optic nerve emerge from our eyes, signal passes through the optic chiasma, then optic tract, then the lateral geniculate body. Then through optic radiation, signal go to the occipital area, which is area 17 in our occipital lobe. From occipital lobe, signals go to the frontal eye field area, which is area number 8. From the frontal eye field area, signal go to the Edinger-Westphal nucleus, which is present in the midbrain. From Edinger-Westphal nucleus, signal go to the ciliary ganglion, then to the short ciliary nerve. Short ciliary nerve causes contraction of the ciliary muscle and that increases the curvature of our lens. Lens becomes globular, its power increases. Short ciliary nerve also gives signals to the constrictor pupillae muscle, which constricts our pupil and meiosis occurs. From the frontal eye field area, signal also go to the somatic motor nucleus of the third cranial nerve. Third cranial nerve is oculomotor nerve. This causes contraction of the medial rectus muscle. That's why the eyeballs converge. A student feels difficulty in reading from whiteboard while sitting in the class. However, he can easily read his book held at a distance of 25 centimeter. Which refractive error the student is most likely suffering from? This is myopia, also called as short-sightedness. What is the cause and treatment of this refractive error? Myopia is a kind of refractive error in which the light focuses in front of retina. This is because the anteroposterior diameter of eyeball is abnormally long. Because of long anteroposterior diameter, images focuses in front of retina, so that's why a blurred image is formed. So we have to send the image back towards the retina, and this can be done by decreasing power of the eye, for which we use biconcave lenses. So when we use biconcave lenses, which are thick at the edges and thin at the center, they cause divergence of the light rays before they enter into the eyeballs. In this way, the point of focus shifts from in front of retina backwards on the retina, and the correction occurs and the vision improves. Next question is briefly describe three errors of refraction along with their treatment. Some important errors of refraction are hypertropia, myopia, astigmatism. We have already discussed myopia. Now let's talk about hyperopia. Hyperopia is also called as hypermetropia or long-sightedness. In this case, light is focused behind retina. This is because the anteroposterior diameter of eyeball is short. Now, in order to bring the image from back of the retina in front, we need to increase the power of our eye, and this can be done by putting biconvex lens in front of the eye. These biconvex lenses used in the spectacles can converge the light rays and increase the power of our eye so that the image from the back of retina moves forward on the retina and the vision improves. Astigmatism is another error of refraction in which the curvature of lens is not uniform in all the meridians. Maybe in one meridian the light is focusing on the retina, but in another meridian light is not focusing on the retina. In myopia and hyperopia there are spherical problems, which mean in all the meridians there is similar issue, but in case of astigmatism the uniformity is not same in all the meridians. For example, someone may be having astigmatism in the horizontal meridian but not in the vertical meridian. This kind of error of refraction is corrected by using cylindrical lenses. Next question is, Miss R is very selective in her diet. From last few months, she is complaining of difficulty to see at night. She is diagnosed to be suffering from night blindness. What is the cause of her night blindness? Because Miss R is selective in her diet, so nutritional deficiency of vitamin A is causing night blindness in this case. Next part of this question is, what will be the role of her treatment in the formation of rhodopsin? When we give vitamin A as injection, this quickly improves the vision of this person suffering from night blindness or nyctalopia. Photochemicals in the photoreceptors, for example, rhodopsin is a conjugated protein. This is made of opsin protein and retinal. Retinal is made from vitamin A. So when we give injection of vitamin A, this binds with opsin protein to make rhodopsin, which is required for normal vision. It's important to note that vitamin A deficiency can also occur if someone is having poor absorption of vitamin A from intestines. For example, someone suffering from malabsorption problem in which chronically absorption of fat soluble vitamins may be decreased. Another question is, 65-year-old man reports to his physician with the principal complaint of night blindness. What is the cause of this disorder? Vitamin A deficiency. Which area of retina becomes impaired? Vitamin A is stored in the pigment layer of retina. So it means pigment layer of retina will be having less stores of vitamin A. And this vitamin A is required for the photoreceptors, rods and cones. So photoreceptor layer of retina, which contains rods and cones, will be having less active pigment rhodopsin, because when vitamin A is deficient, then opsin cannot bind with retinal and rhodopsin cannot be made. And because rhodopsin is present in the rods and rods are more concentrated in the periphery of retina, so periphery of retina will be more affected, which is important for night vision. Next question is, what is Argyll-Robertson pupil? Argyll-Robertson pupil is that pupil which does not respond to light reflex but responds to accommodation reflex. It means that the nervous pathway for both of these reflexes is different. So when we are performing the accommodation reflex, the eyes are showing the changes. But when we are performing the light reflex, pupil is not constricting. This kind of pupil is seen in a person who is suffering from syphilis. And syphilis is a sexually transmitted disease which is caused by a bacterial infection. Next question is, draw a rhodopsin visual cycle. This is also called as retinal rhodopsin visual cycle. When light falls on rhodopsin, which is pigment present in rods, then it converts into some other intermediates, bathorodopsin, then nubirodopsin, then metarhodopsin 1 and then metarhodopsin 2. Metarhodopsin is the activated rhodopsin, which is important in the phototransduction process. This makes us see. This metarhodopsin breaks into scotopsin and all transretinal. It's important to convert these two components, scotopsin and all transretinal, back into rhodopsin so that the concentration of rhodopsin does not decrease in our rods, and we keep on watching continuously. We need an enzyme called as isomerase which converts all transretinal into 11 cis retinal. 11 cis retinal conjugates with scotopsin to resynthesize rhodopsin. Vitamin A, which is already stored in the pigment layer of our retina or in the liver, that is in the form of all trans retinol. This alcohol form is isomerized first into the 11 cis retinol and then it is converted into 11 cis retinal. It's important to note that it's 11 cis retinal which is present within rhodopsin. So this 11 cis retinal will conjugate with scotopsin to make rhodopsin once again. So when rhodopsin is made once again, then the enough concentration of rhodopsin will be there and we can keep on watching continuously. But if there will be no resynthesis of rhodopsin, then we cannot see properly during darkness, and this is what happens in case of vitamin A deficiency. Next question is, what is the mechanism of phototransduction in retina? We have studied that all the sensory receptors are transducers, which means they convert stimulus energy into the electrical change, and phototransduction means light energy is converted into the electrical change in our photoreceptors. How does it occur? When light falls on rods and cones, there is formation of meta-rhodopsin 2, which is activated rhodopsin. That activates a G protein, which in our eyes is called as transducin. Transducin G protein activates an enzyme which is called as phosphodiesterase. This converts cyclic GMP into 5' GMP. GMP is required in the outer segment of our rods to open sodium channels. So when cyclic GMP level decreases by converting it to 5' GMP, then sodium channels close, and then the entry of sodium from the extracellular fluid into the outer segment of rods will be decreased. And when less sodium is entering into the cytoplasm of rhodopsin, that will cause more negativity or hyperpolarization. When there is no light falling on the rods, the sodium channels are open, and that's called as leakage current of sodium or dark current. When sodium channels are open, when no light is falling on the rods, that's called as dark current or leakage current. And because of opening of sodium channels, sodium will flow from out to inside of the cell, and that will create less negativity. So resting potential will be minus 40. But when light falls on the outer segment of rods, the sodium channels close by decreasing the cyclic GMP, and that will increase negativity inside the cell, because positivity of sodium will not be entering into the cell, so negativity will increase inside the cell, and this will create hyperpolarization of minus 70 to minus 80 millivolts. Keep in mind, vision is the only sensation in which stimulation is by hyperpolarization, and this hyperpolarization will decrease the release of neurotransmitter glutamate that will affect the response of bipolar cells and ganglion cells. Next question is, when we move from a bright sunny area outside to a relatively dark room inside, it is difficult to see at first. But gradually our eyes recover and become more sensitive to dim light indoors. Explain the physiological basis or mechanism of this phenomena and make a diagram to elaborate the answer. Another question is, a young man enters in dim light after remaining for many hours in bright light. Which changes occur in the eyes in dim light? Another question is, name the physiological phenomena that makes us see during very dim light. Describe the mechanisms involved. Another similar question is, how do eyes adapt to darkness? Give the significance of dark adaptation. So all these questions are related to dark adaptation. So dark adaptation is a process by which a person is able to see objects during dim light. For example, if you are sitting in a room and you are studying, suddenly if there is power shut down, you will not be able to see anything in the room initially, but gradually you will be able to see something. And same happens when you enter in a dim lighted room like cinema. Initially the person will be blind, will not be able to see anything, and after some time the person will be able to see objects slowly, and this is because of dark adaptation. Maximum adaptation occurs within 20 minutes. Both cones and rods play their role in dark adaptation. Cones are the photoreceptors which are quick to adapt during darkness, then followed by rods. But the adaptation which is provided by cones is much less as compared to the adaptation during dark provided by the rods. There are three important mechanisms involved in dark adaptation. First one is to increase the sensitivity of our rods, and this is by increasing the photopigment rhodopsin. In a well lighted room, rhodopsin is broken down and its concentration is low in our rods. But in a dark room, retinal will be converted back to rhodopsin. Therefore, the sensitivity of our retina will automatically increase in dark condition, because more rhodopsin means our rods will become more sensitive even to dim light. Secondly, pupil dilates in dim light so that more amount of available light enters into our eyes. And thirdly, some centripetal signal from the brain are sent to our eyes, and the photoreceptor cells in our retina, they are excited. So this neuronal excitation mechanism is also important to increase sensitivity of our photoreceptors, and this plays a role in dark adaptation. If you are asked a question in examination how light adaptation occurs, that will be by the same three mechanisms but in the opposite direction. Next question is, what do you understand by protanope and deuteranope? Protanope is a person who is unable to perceive any red light. So it means this person will be red blind. And those persons who are unable to perceive green light, they are called as deuteranope. They are green blind. Next question is, which receptors are responsible for color vision and where are they located? Cones are responsible for color vision. Cones are present in whole the retina but less in the peripheral retina, more concentrated in the fovea centralis and macula lutea. There are three types of cones which are sensitive to three primary colors: blue, green, and red cones. And there are three types of color pigments: blue sensitive color pigment, green sensitive color pigment, and red sensitive color pigment. And only one type of the color pigment is present in one type of cones. Cones are responsible for color vision and also acute, bright, and distinct vision. Next question is, name any five neurotransmitters which are released by retinal neurons. Rods and cones release neurotransmitter glutamate at their synapses with bipolar cells, and glutamate is an excitatory neurotransmitter. Amacrine cells of the retina also produce different types of neurotransmitters, and some important examples are gamma aminobutyric acid, GABA, glycine, dopamine, acetylcholine, and indolamine. All these neurotransmitters released from amacrine cells are inhibitory in nature. So these were important UQs from past papers of UHS. Please don't restrict yourself just to the discussion of these past papers. You are recommended to study thoroughly from the textbook. Thank you so much for watching this video. See you next time with another video.